Dr. Lewcock really came to know to me through a great study that he was doing that was in fact funded by the Philadelphia chapter and led by an academic group, Steve Burden. He doesn't have time to tell us about that today because from Genentech he moved himself to a new company, Denali. Dr. Lewcock has years of experience in industry and really has been pivotal in our programs as a reviewer to critique new programs coming on board. And it is because of that he's here today for our review process that we had the privilege to be able to have him speak today. And he will tackle the challenges of drug development and tell us what it's like to be inside the industry and really how you can get a treatment out the other end and what the challenges are in groups. So thank you for being here.
Thanks, Lucy, for the introduction. And it's really great to be here. I have to say, as a drug developer, it's really easy to get engrossed in the science of what I do in my everyday life. But presentations like this, to get out and see patients and families and things like that, it really reminds me of why I do what I do. And so it's really great to be here. And as Lucy mentioned, so I recently moved to a new company called Denali Therapeutics, which is in San Francisco. And our sole aim at Denali is really to go after neurodegenerative disease. Prior to Denali, I worked for about 10 years at Genentech, where I was initially hired to start an ALS program at Genentech. Before that, we had nothing. Genentech's classically been known as an oncology or cancer biology company. And so it was really great to really be engrossed in the field of ALS drug discovery. And what I want to talk about today is really how I've seen that field evolve over the past 10 years and what I see for the next 10 years ahead, which I think is a brighter future than the past 10 years. And we've grown a lot as a drug development and research community. So just a quick overview of what I'm going to talk about today. That, as I mentioned, what I'll try to do is give you a rough overview of what's going on in ALS drug discovery. Of course, I'm not going to be able to talk about everything, but I'll give you a couple of examples of things that I'm most excited about. And not coincidentally, those overlap with some of the things that Lucy is most excited about. So at least it's good we're all thinking about the same thing. And then I will share one example from the work that we did at Genentech prior to my move to the new company. Then I'll also talk about the current challenges in the field of ALS discovery, and at least my perspective on how we can go about fixing them in the future. And really then talk about why I think I'm excited about what the next 10 years holds for ALS drug discovery, and I'll tell you why. And then lastly, please interrupt me whenever you want. I really enjoy an interactive presentation. I'm happy to talk about anything you guys want to talk about. And my purpose in being here is to inform you guys and tell you things you want to know. So please feel free to raise your hand, or if I'm not paying attention, yell at me. That's fine. Maybe coming up here to be a little too far, but so to get started, then I just want to start with the sobering reminder that drug discovery is really hard, and it takes a really long time. And I think that's important to keep in mind when you hear about a lot of the things that Lucy mentioned, where these are discoveries that were quite literally made in the past 2 or 3 years. And they're very exciting, and I'm going to talk to you about them. But for them to translate into therapies, that can take some time, right? And here's an example of what that looks like. And this is just not for ALS, just generic. And we usually show this to give people an idea of what drug discovery is really like. And so what we have here are from the very earliest stages, the drug discovery in the preclinical stages, through clinical trials to an approved drug, right? And what you can see, the first thing to take away from this is you're talking for every 5,000 or 10,000 compounds that feel like interesting starting points, only 5 of those on average are going to make it to clinical trials, and that equates to only 1 approved therapy, right? So it's a real numbers game, and it's a challenging numbers game. The second part I'll direct you to is the time span involved, right? So when you first start a project until you run a clinical trial, the average time is about 3 to 6 years. then to actually go through phase 1, 2, and 3 clinical trials in order to really confirm that your drug has a function, we're talking on the order of another 6 to 7 years, right? So that means that any project I start today, if I'm really lucky enough to win the numbers game, maybe in 10 years I'll have something to show for it. And that's a big challenge, but it's the reality of the world we live in. And also keep in mind that this is a general chart for drug discovery, not just in ALS, but in all indications. For ALS, these numbers might be even slightly more challenging, right? But this is the challenge that's ahead of us, right? It doesn't mean it's not a winnable challenge. I think every person who gives an ALS talk now needs to have some sort of ice bucket challenge picture in here. And I put it in here because I will say, even from the industry perspective, this made a difference. And the patient community makes a difference. Especially I was at Genentech, it's a big company, there's a lot of things going on, a lot of decisions to make. And the openness and the willingness of the patient community to get involved did make a difference in us heading in that direction. So I want to thank you for that. I also enjoy this slide. I teach a class at Stanford, and I showed them this slide to help them relate to ALS because it has all of the people that they know in there, Zuckerberg and Justin Bieber and the like. But my favorite ice bucket challenge, for the record, is Patrick Stewart here. And he's got his ice bucket, and his whiskey on the side. So that's also an option less taken. But how is that translated then to what's going on in industry? And we really have seen a change in the amount that industry is interested in ALS. And here's some examples of that, which this recently came out in a pharmaceutical industry publication, actually just a few weeks ago. And so I wanted to grab it and show it to you here. Because what it shows is, at present, there's 49 companies working on ALS, which equates to 50 different ALS products in some stage in development, right? And that's a big change from where we were 10 years ago. That's a really fundamental increase. And then when these ALS compounds were broken down by stage, you can see that most of the ALS programs are in what's called preclinical. So that means they haven't reached clinical trials yet. They're still in testing, with a few scattered molecules through different stages of clinical trials. And so to me, I would say part of this high number of programs in preclinical, that's okay. That's kind of what you want to see as a drug discovery person, because not every idea is a good one. And as much as we want benefits for patients, we don't want to take bad compounds forward either. So you want to be able to weed these out. But it's also an indication of how new the interest in ALS drug discovery is. And there's a lot of programs that have come on in the past few years. But it's great to see from the industry's perspective that people are interested. But still, in the past 10 years, we haven't, 10 to 15 years, we really haven't had a lot of success in ALS, and so in developing successful drugs for ALS. And why is that the case? Well, here's my perspective on it. The first is, and this is taking a historical perspective, that we've really had an absence of good drug targets. And the reason why is we still don't understand the mechanisms of disease very well. And we're getting a lot better, but we haven't understood the biology. The second is this thing called the BBB, or the blood-brain barrier. And this is getting a lot more attention recently. What it means is basically our brains have a barrier in them so that a lot of drugs you take won't get in the brain. They get pumped right back out. And it's a protective mechanism to keep stuff out of our brains. But it means when you take a small molecule drug, that it's a lot harder to get it into your brain than it would be to get it to other organs in your body. And so that's a challenge for us as drug developers. The next is, as Lucy mentioned, biomarkers. And I think this is of particular importance for ALS, and I'll talk a little bit more about what I think we can do with biomarkers to make a difference in the future. And then lastly is what I would define as core translational medicine. And what that means is, we can have the models, we can have the assays, but we need to really utilize them effectively to give us meaningful information. And a lot of companies, I'm going to say that companies can be quite guilty in this, in pushing their product too far ahead before they have the data to say it's worthwhile. The good news is I think all of these are changing now, and I'm going to get into some of that in the last part of my presentation. But what I'll first start with is a couple slides on what we did back at Genentech when I started working on ALS in 2006. And at the time, we were so perplexed by the biology. A lot of the new genetic hits, the C9orf72, the TDP43, they hadn't been discovered yet. We really didn't have anything. But what we did have was we knew that there were certain patients with familial ALS that had genes that cause ALS, things like SOD1, and that was known very well, and there were great models for that. We also, there was mounting evidence that there were some environmental causes of ALS, and one example would be veterans of the first Gulf War that came down with ALS at a much higher incidence than the general population. But in reality, these were very small parts of the overall ALS population. And the vast majority of ALS patients had sporadic disease, where it was an unknown cause, but likely some combination of genetic factors and environmental factors that led to disease. So we reasoned that even though there could be these diverse causes of disease, that patients with ALS did share a common phenotype, meaning they got common symptoms. They had motor neuron loss, they had motor weakness, they had paralysis. And so we reasoned that if these all filter down into a common phenotype, common symptoms, that there may be common pathways that lead to this degeneration event, regardless of the initial cause in ALS. And so these were the pathways we decided to take a look at the time. And the way we started doing that was by culturing neurons in a dish. And so I'm going to show you a little movie here from Neurons in a Dish. And what you're looking at these are what's called axons, or these are actually the nerve fibers. So if your neurons in your spinal cord project out to control your muscles, there are these axons or fibers, and these are them growing in a dish. And we created an experimental setup where we could watch them degenerate and then try to find things that stop that degeneration, hoping that this would be a common mechanism of degeneration, or a common mechanism. And so what you're looking at here is a time-lapse movie, so it goes over the course of 24 hours, but it's sped up so you don't have to sit here all night. And what you can see is the axons are at first fine, but then they start to get these blebs or varicosities in them, and those are similar to what you see in disease. And then over a very short time span, they just completely disintegrate, right? And so, I mean, this is a very, very low complexity version of what we thought might be happening with degeneration in patients. And so we took these initial observations and really through work from a number of my colleagues and a number of years of work, we identified this kinase, dual leucine zipper kinase, or DLK. And it seemed when neurons lacked DLK, when you remove this gene in experimental systems, that when these axons here were completely degenerated, that these DLK knockout axons, the nerve fibers, were still intact, right? And so this seemed like something exciting to go after, and this is just a pathway by which DLK signals in a cell. And so to fast forward a number of years, we tested it in the SOD1 model of ALS because that was the best model available at the time, and we knew that motor neurons degenerated in that model. And this is just some representative results from that. And what you can see is what was pretty encouraging in that, so these are spinal cords, and all these dark spots are motor neurons in the spinal cord of these mice. So normal mice, whether they have DLK or they don't, the motor neurons look fine. SOD1 mice at about mid-stage disease, you can see it, how many motor neurons they've lost here, right? But when you lose DLK expression in these mice, a lot like we saw on the dish, we're able to rescue some of these motor neurons, but they're still alive in this stage of disease. And then when we looked at survival in the SOD1 mouse, that there was some benefit of losing DLK expression on survival as well. So this was an encouraging project, a lot of years of work, and we did develop some DLK inhibitors at Genentech, which are currently in phase 1 clinical trials. So the very earliest stages of testing, just single doses, just to see if it's tolerated by patients. But it is an example of how long this takes from a project that started in 2006 with the hypothesis based on the data that we had in 2006. It started clinical trials in 2016, right? So it was quite a long time. So let's then move on and take a fast forward to 2016. Let's go to now, right? And say, what have we learned in the past 10 years? And one thing we've learned is if we look at other places where we've been developing drugs, other areas have been a bit more successful. And one of those is oncology or cancer biology, right? And that's shown here where, if you look in this chart, the red line, there's no way you can read these axes, I'm sorry, but the red line is showing the number of cancer-related deaths over the year, right? So this is from 1990 to 2015. And this is the number of cancer survivors, right? So you can see a decrease in death and the increase in number of cancer survivors. So why have we been successful in cancer? Well, what happened in cancer was people identified oncogenes, or genes that cause cancer in cancer patients. And then they targeted drugs to those oncogenes, right? They went directly after them. And that was a very targeted approach. It went after the mechanism that was a primary driver of disease. And that was a lot different than we were thinking about ALS or other neurodegenerative diseases at the time because of the poor understanding of biology. And so, what they were able to do is really say you could go from oncogenes to the cellular pathways that define them, define targets and therapies. Then, when you go into clinical trials, because you know how your drug works, you can measure what's called target engagement. And target engagement means a measure that your drug is doing what it's supposed to do in a patient, right? And you can usually do that very early on in the trial. And you define that your drug hits your target, and this allows you to pick a dose and know that you have confidence that when you enter longer trials, that you're actually going to be effective. And then lastly, again, back to biomarkers, where you have to pick a particular patient population that you think is going to respond to your drug. And that may not be all of ALS patients. It may be one small subpopulation of ALS patients that's defined by a gene that's mutated. And to go after those patients gives you a better chance of success rather than try to get them all in one lot. And so, the question that we've come to is, can we then now, moving forward, apply this oncology mindset to neurodegeneration and apply it to ALS? And I'm optimistic that the answer is yes. And the reason why I think that is, and Lucy mentioned some of this, this was meant to be too small for you to try to read it, but these are all of the genes that have been identified as potential causes or risk factors for ALS. Right? For a long time, we didn't have a lot, but now we have a lot to work with. And the second part of that is a lot more work on biomarkers, a lot of it funded by the ALS Association. And biomarkers can come in a number of different flavors, but all of them are going to be really helpful for running successful clinical trials. And I'll talk about that in a little bit more detail. So Lucy talked about this a little bit, but to get back to why are biomarkers so important? And to give a graphical example of that, I'll just show you, this is data from an ALS clinical trial, right? And this is what I think you guys are used to seeing, which is unfortunate, that you see a Kaplan-Meier plot and you're looking at survival of patients over time. And what you see, unfortunately, is that there's no separation between treated patients and placebo, right? And this is the data that you usually see, right? But when you break that data down, and instead of looking at a population, you look at individuals with ALS, and you ask how fast do certain individuals with ALS progress. What you see is something more like this, where some ALS patients progress very quickly, and some ALS patients progress much more slowly. And on average, you get a curve like this. But to try to find what's a real effect of a drug and what's just different progression rates between different people is a real challenging prospect, right? So one of the things that we could do with biomarkers, and there's a lot of effort on this, and I think it's well worth it, is to say, how can we narrow down the spread and identify a certain population of patients that might be expected to respond to our drug, or might progress at more of a homogeneous rate such that we can measure the effect of our drug more easily. And then the second thing that we can do, as I mentioned before, is measure that target engagement. So very early on, you treat a patient with your drug, and you can say, I know that this drug is doing something in a patient. And far too many times we've gone into long trials where you might treat a patient for 12 months, but there's not a clear idea whether that drug is actually doing what it's supposed to be doing. And so this is what biomarkers can really do. The second part is human genetics, and Lucy talked a bit about human genetics, and I'll show this, this different graphic, which I really like because it incorporates time. I guess yours incorporate time as well. Mine paints a more pessimistic picture up until, 2008, where basically we had mutations in SMD1, and we've known about mutations in SMD1 since the 90s, right? But until, for a long time, SMD1 mutations were all that we knew about the genetics of ALS. And up until very recently, and now you're looking here at 2008 and beyond, we identify mutations in this TARDB, that's TDP43, that's the same gene. We scientists like to give things multiple names to confuse people, sorry about that. And FUS and BCP, and then the other one that Lucy mentioned and I'll talk about as well is C9orf72. So we've really made very big gains in our understanding of the genetics of ALS. And if you look on this axis here, what you're seeing is the percentage of ALS that's explained by these genetics, right? So for sporadic ALS, there's still a lot that's unknown, a large percentage of ALS for which no genetic cause has been defined. But with familial, we're getting close to understanding, the majority of what causes familial ALS, so ALS that runs in families. But we're getting closer, and we at least have some tools in the genetics to work with now. And so this is just another way to look at that, where about 68% of familial ALS, the genetics that define it, have been found. With sporadic, we're only at about 11%. And that doesn't necessarily mean that all 90 or 89% is going to be explained by any one other gene. But we are making progress. And so what I want to do now then is just to talk about two of the same examples that Lucy's excited about, which I'm excited about, C9orf72 and TDP43. And what at least I think about them from a drug discovery perspective. So C9orf72, Lucy talked about it a little bit. Funny name, C9, it just means it's on chromosome 9, open reading frame 72. So they kind of randomly assigned genes names if we hadn't given them a name yet. And what you get in this gene is what's called a repeat expansion. So in our DNA, there's naturally repeated sequences, and then every once in a while, when our DNA gets duplicated, it skips, and that repeat expands and gets longer. So that repeat turns into from 3 repeats to 100 repeats, let's say. And so what does that do? Well, this repeat turns into what's called an RNA molecule, and that RNA molecule, because it's just a repeat, it folds up on itself and gloms up, right? And this is one of the first things that was identified from the C9orf72 gene. The second thing it does is this repeat sequence in the DNA makes a repeat protein, right? So DNA makes proteins in your cells, okay? And it's a code to make the right protein. But if you have a repeat code, you just make gobbledygook. That's the same thing repeated over and over again. And that doesn't make any sense. And what happens with that is that gloms up in your cells as well. And when you look at patients with ALS that have the C9orf72 mutation, you see gloms of these proteins that are made from this repeat in the neurons, right? And you can also find it in the cerebrospinal fluid of those patients, right? But they think that this is one of the causes of disease in C9orf patients. From a drug discovery's perspective, this type of thing and this type of understanding, all of which has come since 2011 till now, so in the very recent past, is really exciting. Because what it gives us is first a link to human genetics. And the reason why we like that link to human genetics is we can say we know that this is important in human disease. It's not just an artifact of what we model in a dish, and we can identify patients that have the mutation. The second part is we can use these gobbledygook proteins that get made as a biomarker. We can use them to look at identifying patients that have it present for enrollment in a trial. Or we can also say, if we are trying to get rid of it, as Lucy was mentioning with the Ionis Biogen collaboration, that this could also be a measure that our drug is working early on, so we can have confidence to go into end-stage trials. I think what we're lacking at this point is the true therapeutic hypothesis, meaning we know that these are the problems, how are we going to fix them? And that's really part of where we're at right now with ALS drug discovery, because some of these genetic factors are so new. But it's also why I think the next 10 years is going to be a very exciting time for research and drug discovery, because now we really have this to act on, where for the 10 years before that, we really didn't have even these tools available. Lastly is TDP-43. So TDP-43 is, it doesn't really matter the structure of the protein, just to know that this region at the end is where all the mutations that cause ALS are found, right? And so I try to give the very high-level overview of what might be happening with TDP-43. So I came up with 3 interesting facts about TDP-43. And so maybe this will make you more confused, or maybe it'll be interesting, probably some combination, depending on who we ask. So first, TDP-43 is an RNA binding protein, so it binds this stuff called RNA in the cell. That's its normal function, right? And it has this sticky end on the end. It's naturally supposed to be sticky, and it's part of the way the protein works, okay? And TDP-43 is not the only, it's the most well-known of these RNA binding proteins that's implicated in ALS, but there's more than one of them. There's been others identified since. And like FUS is an example of another RNA binding protein. And why that's interesting to me as a drug discoverer is because when you have one hit and you're unsure what's going on, it's harder to put together an idea of how to make a drug. But if you have multiple ones that are doing the same thing, it really points to a pathway that's misregulated in disease. And that's what we really need to hook into for drug development. And then lastly, where all these mutations are in TDP-43, and this gets to some of the questions before, it makes that sticky end of TDP-43 more sticky. Okay? And all of the mutations in all of these RNA binding proteins that cause ALS are all in the region of the sticky part of the molecule. So why does that make sense? Because now if you start to look at pathology, you look at aggregates in the brains of ALS patients, what you see are this sticky molecule now has irreversibly stuck together and formed aggregates in the neurons. And so you don't, and why it's exciting for drug discovery is, you know, the sticky molecules aggregating together, it starts to make sense from a pathway perspective. The mutations in TDP-43 or other RNA binding proteins are very rare, but you don't have to have those mutations to have these aggregates in your brain. If you have almost 100% of sporadic ALS patients, if you look at their brain after they pass, you find that they have these aggregates present. So it's present in almost all of disease. It really points to a pathway that might be affected in a large percentage of patients. So this is something that, again, the therapeutic hypothesis, what to do about it, we still have a lot of work to do, but it's a really exciting starting point that we're really working on yet.
So it occurs naturally, it's sticky, but when it becomes stickier than it should be, does that cause a problem, right?
That's the hypothesis, that's the idea.
One makes it more sticky.
Yeah, and so, well, the problem, I think, is slightly even more complicated than that. So what makes it more sticky is the mutations, right? But then the question is, why is it sticking together abnormally in disease? Why does that happen sometimes and not in other people, right? And if you can understand the pathways that lead it to stick together abnormally, then those are the things that you want to develop drugs against. Make sense? Okay. Questions?
We know there's clot-busting drugs for heart and stroke. Are there protein-busting drugs in the pipeline to get rid of this?
That's a great question. The question, just to repeat it, is, are there protein-busting drugs? And I think this is an idea that many people have had, and intuitively it seems like a really good idea. The key is to be able to do that specifically for these proteins, but not other proteins. So part of normal protein function is for them to interact with each other. And so you need to be able to have a drug that blocks what I'll call pathological or disease-related clumping, but doesn't affect the normal interaction. And that's harder to do. But there are approaches now, there are some new approaches. None of them have reached clinical trials yet, but some therapeutic antibody-based approaches that are headed in that direction, and I think they're very interesting. Okay, so back to my somber reminder of the challenges of drug discovery. It takes a long time, but I really think that we're getting there, and the next 10 years I'm really excited about, based on some of these interesting new starting points, that we have a much better opportunity to develop successful therapeutics. So just to bring it to a close quickly, I think that there are some exciting therapeutics for ALS in the clinic, and Gingy's going to talk a little bit about those in the next presentation. But I'm hoping that there will be more to come in the coming years. We've started to identify some of the genetic causes of ALS, and I think that's going to give us a lot of anchor points as a drug discoverers. But still, about 80% of disease has unknown cause. The next generation is in the works, and that's really driven by our understanding of disease mechanisms, and the term that Lucy used, the idea to do personalized medicine. That the idea that treating all patients with ALS, there may be certain instances where that works, but there may be other instances where you have to identify a certain population of patients based on a trait, based on a genetic trait, based on how their disease manifests, and go after them with a specific therapy, and that it might not be as uniform as one disease. And that's what's really been successful in oncology, and I think that's where we need to go. So thank you for your attention, and I'm happy to answer any questions.